Hi, Assalamualaikum everyone. Now we're going to present our CHN301 video presentation about tyrosine. First of all, let me introduce our group member. First, Ilis Naris Kairin binti Kaironizat, and student ID is 2022496722. Second, Nur Aina Jiha binti Fazli 2022696624. Third, Iman binti Zaini 2022880726. And lastly, my name is Siti Nur Umaira binti Muhammad Abdul Qadir. My student ID is 2022896826.
First, for the introduction, mammals synthesize tyrosine from ancestral amino acid phyllophenin PHE, which is derived from food. We can find tyrosine in soy product or chicken, turkey fish, peanut, almond, and avocado. The structural formula of tyrosine is C9H11NO3. The IUPAC name of this tyrosine is 2S2 amino 3,4 hydrophenylpropanoic acid. The common name of tyrosine is 4-hydrophenylalanine. For the functional group is tyrosine. But for the homologous series, we determined that there was no tyrosine in homologous series because tyrosine is one of the amino acid group.
Alright, so do you guys know what the physical and chemical properties of a tyrosine are? First, we have molar mass. The molar mass of a tyrosine is 181.191 gram per mole. And as for its appearance, it appears as a white solid colored crystal powder. And for its density, we have 1.34. And as for its solubility, it is less soluble in water. As we can see, 1 mole of hydrochloric acid equals to 25 milligram or milliliter of water. So as for its pH, it is 6.5, which is considered as a weak acid. And for its odor type, it is odorless. It didn't smell. And as for its melting point, it is above 300 degrees Celsius or around 340 degrees Celsius. So lastly, we have the boiling point, which is 314.29 degrees Celsius. So let's move on to the chemical properties of a tyrosine. So we would say that tyrosine is actually considered as a weak acid because it can donate a proton to a solution, but it doesn't mix well with water like strong acids do. So this happened since phenyl group exists in its structure. For hydrophobicity, it is hydrophobic as it can influence the location within proteins and its interaction with other molecules. And as for the hydrophobic interactions, it is playing a very crucial role in the folding of proteins and the formation of cell membranes or the assembly of DNA. The reactivity of a tyrosine is very reactive because it is participate in various biochemical reactions due to its chemical structures. And lastly, halogenation. It is possible to undergo halogenation to alter its structure, but also under certain conditions such as high concentration of the acid solution, such as hydrochloric acid.
So for the uses of tyrosine, the uses of tyrosine can be seen in phenylketonuria treatment, or PKU. Phenylketonuria is a rare metabolic disorder characterized by impaired conversion of phenylalanine, PHE, to tyrosine. If left untreated, the resultant accumulation of excess blood, PHE, can cause physiological, neurological, and intellectual disabilities. Therapies that are in PKU treatment are dietary treatment, tetrahydropterin as enzyme enhancement therapy, enzyme therapy, cell-directed therapy, and gene therapy. Gene therapy uses tyrosine. Studies in PKU mouse models have shown that gene therapy can be successfully delivered to non-hepatic tissues such as the muscle. In fact, the insertion into the muscle cells of vectors containing the necessary genes for PAH and tetrahydrobiopterin synthesis resulted in a system that could convert phenylalanine into tyrosine, mimicking the role of hepatic phenylalanine metabolism. Other than the phenylketonuria treatment, tyrosine can also be seen as targets for anti-cancer agents. The role of tyrosine kinases in cancer molecular pathogenesis is immense, and recently kinases have come in vogue as potential anti-cancer drug targets. As a result, a couple of anti-cancer drugs are in the market. The complexity and the number of tyrosine kinases have greatly increased with the sequencing effort of the Human Genome Project, thus providing more opportunities for drug discovery. Recent understanding of the molecular pathophysiology of cancer have highlighted that many tyrosine kinases are found upstream or downstream of epidemiologically relevant oncogenes or tumor suppressor, in particular, the receptor tyrosine kinases.
So next, for the chemical reaction, first, the reaction of decarboxylation to theramine. Tyrosine is converted to theramine by the enzyme tyrosine decarboxylase, which catalyzes the removal of the carbonyl groups COOH from tyrosine, producing the theramine and releasing carbon dioxide CO2 as a byproduct. So here is the example of the structure. So for the second reaction is reaction of L-tyrosine converted to L-dopa. L-tyrosine is converted to L-dopa in a single step by the enzyme tyrosine hydroxylase. This enzyme uses the tetrahydrobiopterin and oxygen as a cofactor. The reactions add a hydroxyl group to the meta position of the benzene ring in tyrosine, producing L-dopa. Tetrahydrobiopterin is oxidized to dihydrobiopterin during this process, and water is formed as a byproduct. This conversion is crucial for the biosynthesis of dopamine and other catecholamines, which are essential neurotransmitters. So here is the example of the structure. In conclusion, tyrosine is an essential amino acid critical for synthesizing neurotransmitters such as dopamine, norepinephrine, and epinephrine. It serves as a precursor in various metabolic pathways, including the production of thyroid hormones and melanin. Tyrosine enzymatic conversions facilitated by specific cofactors like tetrahydrobiopterin and pyridoxal phosphate underscore its role in maintaining neurological function, hormonal balance, and pigment formation.
That's all from us. Thank you for watching. Bye.